going to go ahead and introduce our next speaker, Professor Toru Okamoto. You may go ahead and start sharing your screen. All right. So Professor Toru Okamoto is a professor in the Research Institute for Microbial Diseases in Osaka University. He also graduated from the Department of Biotechnology in Osaka University and received his PhD from Osaka's University School of Medicine. He did his postdoc in the Water and Eliza Hall Institute in Melbourne, where the main research focus of his lab was on pathogenesis of hepatitis viruses such as hepatitis C virus and hepatitis B, and mosquito-borne flaviviruses such as Japanese encephalitis virus, dengue virus, and Zika virus. And he's going to be talking about the development of an engineered ACE2 decoy for the treatment of COVID-19.
Okay, thank you very much for your introduction. Can you hear me? And the slide is okay?
Yep, looks good.
Okay, so I would like to thank you very much for to all organizers for giving me opportunity to introduce our research today. My name is Toru Okamoto from Osaka University in Japan. Today, my talk is about development of an engineered ACE2 decoy for treatment of COVID-19. So infection SARS-CoV-2, SARS-CoV-2 infects into the respiratory organs, such as showing here, and many cases of this virus infection eventually causes severe pneumonia. This is a scheme of a viral life cycle of SARS-CoV-2. SARS-CoV-2 can bind to specific receptors such as ACE2 and then enter the cells. After internalization of the virus, viral RNA is released from viral capsid, and released viral RNA translates viral proteins. Viral proteins form a replication complex showing here to propagate viral genome. So this viral replication complex propagate viral genome. Viral genomes are consisted of genomic RNA and 4 different subgenomic RNA. And these viral RNA translates viral proteins to form viral particles and release outside the cells. So today, my talk is focusing on this entry step of virus infection. SARS-CoV-2 recognizes host proteins called human ACE2 as a receptor. Viral spike proteins of SARS-CoV-2 specifically binds to ACE2, and then viruses enter the cells. Interaction between viral spike protein and ACE2 is very specific and possesses high affinity binding. In this study, we try to design ACE2 possessing much higher affinity to spike protein called as ACE2 decoy. We examined how effective of the ACE2 decoy in viral propagation and treatment of COVID-19. To generate high affinity ACE2 decoy, we performed error-prone PCR. The cDNA of ACE2 was introduced random mutation to generate ACE2 expression library. The ACE2 library was expressed in 293 T cells and incubated with GFP fused with SARS-CoV-2 spike protein. GFP high-expressing cells were sorted and identified their mutations. Finally, we identify 3 different cDNA clones to enhance the affinity to spike protein, called 3N39, 3J113, and J320. We then try to identify essential mutation to enhance the affinity to spike protein. We can see here, among the several mutations, these 4 mutations of each ACE2 mutations were necessary for enhancement of affinity to spike protein. Finally, we got 3N39 version 2, which is about 30 times higher affinity to spike proteins compared to wild type. Next, we try to understand how 3N39 enhanced binding affinity to spike protein. X-ray analysis suggested E35K mutation, shown here, generated a new interaction with Q493 of spike protein. In addition, A25V mutation and F79 mutations enhanced hydrophobicity to interact with spike protein, as shown here. So ACE2 has originally enzymatic activity to regulate blood pressure and so on. Previous ACE2 structure was solved as open conformation showing here. This open area was thought to bind to ACE2 substrate. Interestingly, our ACE2 structure was formed as closed conformation shown here. So we identify valine 343 and serine 128 was the closest in this structure. Therefore, we mutated 343 and 128 to cysteines to generate SS bonds to keep closed conformation of ACE2 protein. So we found that introduction of SS bonds completely lost their enzymatic activity shown here without reduction of affinity to spike proteins. In addition, we also show that mutation of cysteines enhanced protein stability as shown here. So next we examine the antiviral effect of ACE2 decoy. To produce efficient recombinant proteins, ACE2 decoy was fused with FC region of human IgG. Then neutralization of pseudovirus was observed by 3N39. IC50 value was 56 nanogram per milliliter compared to 24.8 microgram per milliliter of wild type. We also confirmed ACE2 decoy can inhibit viral propagation by using authentic virus, suggesting that our ACE2 decoy have antiviral effect. Then we next examine the effect of ACE2 decoy on propagation of SARS coronavirus 2 variants, VOCs. We collected these infectious viruses containing BA2.75, BA5, and BA.4. We found that our ACE2 decoy 3N39 showed neutralizing activity against all VOCs, as shown here. Next, we examine the effect of ACE2 decoy in vivo. Syrian hamster was infected with SARS coronavirus 2, and ACE2 decoy were treated 2 hours post-infection. We can see here, CT analysis data shows control hamsters develop pneumonia. In contrast, treatment with ACE2 decoy improved lung pathogenicity. H staining and spike protein IHC data showed control FC treatment development severe pneumonia. However, ACE2 decoy treatment improved lung pathogenicity. Again, amounts of viral titer and viral RNA was also improved by treatment with ACE2 decoy. In addition, expression of inflammatory genes such as IL-6 and IP10 was also improved by treatment with ACE2 decoy. These data suggested that ACE2 decoy is effective in vivo. So next we examine the effect of mutation of ACE2 decoy on T-cell activation. We asked this project to Proimmune company and wild-type ACE2 peptide and all relevant mutant peptide were incubated with various HLA-derived CD4 T cells. We can see here no activation of CD4 T cells was observed by treatment with wild-type peptide over ACE2. Among the mutant peptides, we found that F79 mutant peptide were mutant stimulated particular T cells as shown here. Therefore, we mutate F79 to wild type as a 3N39 version 3. Then we performed another screening to identify alternative mutation of F79F mutation. After screening, we identify T92Q mutation could compensate F79F mutation. And T92Q mutation was contained J320 and showed no activation of T cells. Therefore, we decided these 4 mutations containing T92Q mutation instead of L79F mutation as 3N39 version 4 for our final ACE2 decoy product. We next examine the effect of ACE2 decoy on using a monkey model. Cynomolgus monkey was infected with SARS coronavirus 2 and treated with ACE2 decoy 1 day post-infection. After 7 days, we collected tissues. Control monkeys shows inflammation and detected infectious virus showing here. In contrast, inflammation was improved, clearly improved, and no infectious virus was detected by ACE2 decoy treatment. Our data suggested that ACE2 decoy can improve COVID-19 in monkey model. So finally, we examine the possibility of emergence of viral escape mutants. We use recombinant VSV possessing SARS coronavirus 2 spike protein instead of original G protein of VSV. We incubated wild-type ACE2 decoy, wild-type ACE2 and ACE2 decoy or monoclonal antibody beltelovimab with recombinant VSV. We found treatment with wild-type ACE2 or beltelovimab monoclonal antibody generated resistant virus after 7 times passages. In contrast, ACE2 decoy did not show any emergence, any escape mutation as shown here. These data suggested that high-affinity ACE2 decoy could overcome emergence of escape mutations. I would like to summarize our data. So compared to monoclonal antibody using in clinical use, our ACE decoy can be effective to all VOCs. And our ACE decoy is effective to other salvegoviruses. And also our ACE decoy showed no escape mutations. Compared to other ACE decoy previously reported, our ACE decoy could confirm antigenicity, and also our ACE decoy is stable and showed no enzymatic activity. So finally, our ACE decoy can confirm effective in vivo, including monkey model. So finally, I would like to thank our collaborators, especially Atsushi Hoshino and Junichi Takagi, Kyoto Prefectural University of Medicine, and Osaka University Institute for Protein Research. I and these two collaborators performed this research. And also my laboratory member, Tatsuya, Yumi, Mari, and Akira performed an experiment in my laboratory. And I would like to thank my collaborators. And thank you for your attention. I'd like to discuss, and I'm very happy any comments.
Great. Thank you so much, Professor Okamoto, for that very excellent talk. I'll open the floor to questions now.
Yeah, let me ask one question. That very nice talk. Thank you very much for your nice talk. Can you hear me now?
Yeah.
Yeah. Actually, although you tested the antigenicity of your ACE2 protein, I'm curious. In monkey model, the monkey developed the neutralizing antibody against the ACE2 protein. Have you ever checked the antibody titer?
Yeah, actually we use the human ACE2 fused with human IgG. So if we inject our monoclonal our ACE decoy into the monkey produces the antibody. But compared to other human monoclonal antibody, our antibodies amount of the antibody produced by our ACE decoy is very equivalent compared to other human antibody in monkey model. So I think it's not that our ACE decoy has a big. I don't think this ACE decoy stimulate many immune cells to produce the antibody, I think.
All right. Well, thank you again. Thanks very much.
Thank you.